Breaking the Wall of Endometrial Cancer, Honrado, Falling Walls Lab, Berlin-Adlershof.
Endometrial cancer is the most common gynecological cancer. Every year, it's responsible for more than 400,000 new cases and close to 100,000 deaths. And worryingly, it also has an estimated incidence rise of more than 50% worldwide by 2040. In many endometrial cancer cases, the cancer cells trick our immune system by using specific proteins to turn off the checkpoints that the immune cells use to know when to trigger an immune response and kill those cancer cells. The good news is that there are already available immunotherapy options that solve this issue and are highly effective at treating the cancer. The bad news is that there are no easily implementable methods to identify which patients will actually require this treatment. Currently, this assessment is done by extracting tissue samples directly from the cancer site and checking for what are called microsatellite instabilities, or MSI, which are the lesions known to occur in specific areas of the cancer DNA. So the walls we are currently facing are the necessity to use this type of tissue samples because they are highly limiting in terms of disease relapse evaluation, long-term checkup, as well as the methods to assess these MSI, as they typically rely on very expensive and lengthy processes, such as PCR and immunohistochemistry. Our new approach is based on using extracellular vesicles to collect the cancer DNA. Extracellular vesicles, or EVs, are small particles that are shared by all cells in our body, including the tumor cells, that carry with them cancer DNA. These EVs can be found in a simple blood sample, so making recurrent disease checkup a much simpler process. In our approach, we use a microfluidic device to isolate these EVs from patient plasma samples in a faster manner than current methods. And this is because these microfluidic devices, think of them as a little bit of a laboratory inside of chip, where thanks to the reduction of the microscale of the device, all the reagents and the processes needed for EV isolation are much faster and efficient. After the EV isolation, we then collect the DNA from these EVs and we perform the MSI evaluation using spectroscopic methods with what we call nanostars, which are basically gold nanoparticles that we can coat with DNA that specifically targets the cancer DNA and will tell our biosensor if there is MSI or not, thus informing a clinician if a patient actually requires the immunotherapy treatment or not. By integrating all of these steps into a single automated device, we will be providing a sample-in, answer-out system that is not only faster, but cheaper than current methods, and we will be thus finally breaking down the walls in endometrial cancer therapy. Thank you so much.